Mycoplasma. One of the most common contaminants present in cell cultures, this self-replicating prokaryote is estimated to contaminate up to 35% of all continuous cell lines. Lacking a rigid cell wall, they are not affected by pen-strep antibiotics and are too small to be trapped by media filters. They are dependent on their hosts for nutrients and negatively affect any assay the cells will be used for. Contamination can come from anywhere, but most commonly from not following proper aseptic techniques. Most importantly, always wash your hands. AKRON's Mycosolutions detection kit is designed to detect the presence of mycoplasma contaminating biological materials, such as cultured cells. There is no need to prepare probes or to calculate DNTPs or buffer concentrations, as everything comes in a ready-to-use, optimized PCR mix that detects a wide variety of mycoplasma contamination. If any contamination is present, it will be clearly visible on the agarose gel. To make your 2% agarose gel, measure 1.2 grams of agarose and add it to 60 milliliters of TBE running buffer. Add a little buffer to an Erlenmeyer flask. Then add the agarose and the remaining running buffer. Swirl it and heat it in the microwave until it boils. Once it's cooled slightly, add 2 microliters of ethidium bromide. Once the solution has cooled a little bit more, you can pour about 30 milliliters into your gel casting tray. Notice how thick the liquid is. Add your comb and let the gel cool. First things first, we need to transfer some of our cells into tubes so they can be lysed and then run on the thermal cycler. Here we are taking 1 milliliter of cell supernatant and spinning it briefly in a microcentrifuge. Multiple samples may be aliquoted and spun down at the same time. Transfer the cell suspension into a fresh Eppendorf tube, making sure to note the sample you are working with on the tube and in your lab notebook. And remember, it might be contaminated. Place the sample or samples you are working with into the large tabletop centrifuge and set the centrifuge for 15,000 times G and a runtime of 10 minutes. Any nucleic acids will be pelletized in adhering to the wall of the tube, so you may decant the supernatant by inverting the tube. Set your micropipette to 50 microliters. A lysis buffer that comes with the kit should be kept on ice. Take 50 microliters of the buffer and resuspend the pellet by pipetting, not by vortexing. The result should be 50 microliters of clear solution for each sample tube that you will be testing. Use a water bath or preferably a dry heat block and set it to 95 degrees. Make sure that it's at temperature and insert your tubes for just 3 minutes to lyse the cells in the buffer mixture. Now it is time to load our tubes with everything we need for the PCR reaction to take place. Simple. Each of the 4 samples we will be testing for contamination will get exactly the same mixture. Here, we are first loading each of the PCR reaction tubes with 35 microliters of deionized water. Next, 10 microliters of the reaction mix that has the primers and DNTPs necessary for PCR to occur will be added. Make sure to change the pipette tip for each tube to avoid contamination of the reaction mixture. The last step is to add 5 microliters of your sample to the tube. Make sure to note which sample tube is paired with the reaction tube as you add the sample in case you need to identify the sample at a later time. And again, it is critical that you change your tip between each sample that you load. You have all your samples prepped, but you still need to make a tube with your positive control. The positive control will show that the PCR reaction worked and will produce a 270 base pair band that will be visible on our final gel. If you want to feel like a real biotechnician, now is the time. We are about to perform a technique known as PCR, polymerase chain reaction, something only dreamed about a few decades ago. Each reaction tube will be heated and cooled in sequence for 40 cycles until billions of copies of target nucleic acids are produced. All of the cycling protocols we use are pre-programmed in the thermal cycler. If they weren't, it is easy to add one, but in this case, we simply scroll down to select it. Viewing the protocol shows us the target temperatures and the number of cycles that will be run. Remove the 8-toothed comb from the gel and move the gel and casting tray into an electrophoresis boat. You should still have TBE buffer left over from making the gel, and you will pour this over the gel and into the reaction boat until the gel is barely submerged. About 250 milliliters should do it. It is time to load the wells. With a fresh tip, add 6 microliters of the O-gene 1 kilobase ladder to the first lane. Lanes 2 through 6 will be loaded with 20 microliters of your samples and the positive control. You choose those lane assignments. End by loading a duplicate 6 microliters of the gene ruler to the final well. This will be the standard you use to measure the fragment size on your final gel, and having it on both sides of your samples makes reading the gel easier. Secure the top, making sure the positive terminal is farthest from the wells, and plug the leads into the power supply. Set the unit to run for 100 volts for 45 minutes, and press start when ready. After the run, the gel will look the same as it did when we started, since no loading dye was used. But during the run, any amplified product will have moved through the agarose lattice toward the positive terminal. Imaging the gel on a UV transilluminator will cause the ethidium bromide to fluoresce. Wherever there are concentrations of nucleic acid, a band will appear. We will be looking for bands that show us 16S rRNA has been amplified. If we see it, our samples likely have mycoplasma contamination. The gene ruler allows us to measure what we're looking at. Comparing it with the gel, we see there's only one band at 270 base pairs, and that is our positive control. Primer dimer, it's a happy day. We don't have contamination. Mycoplasma is a common contaminant in labs around the world. It is especially pernicious because it's immune to commonly used antibiotics and also passes right through media filters. Look for signals of contamination like color changes in media or poor health of your culture. If you suspect contamination, use AKRON's Mycosolutions Mycoplasma Detection Kit to know for sure. And don't forget the #1 rule of aseptic technique. Wash those hands.